The second wave of bioinformatics is the gene expression wave. In 1977, a technique called northern blot was invented in order to measure the expression of a single gene in a cell condition. This is an experiment that also took a lot of effort to do. In 1995, new technology was developed at Stanford, which used microarrays to measure hundreds or thousands of genes at a time. I remember my undergraduate research advisor came back from a conference in 1996, and he was so excited. Basically, you can use a cellulose membrane and have hundreds of tiny dots or probes, each probe measuring the expression of a single gene. Therefore, using this microarray, you can measure many genes in a specific condition. And so in this experiment, usually you have cells grown in different conditions or samples taken from different tissues or different conditions. For in this case, it's normal cell versus cancer cell. You isolate the RNA and fluorescently label the RNA or convert them into cDNA, fluorescently label the cDNA from two different conditions into different fluorescent colors. And then you mix them together and hybridize this onto kind of this initially was a cellulose membrane, later on was a glass slide. And the glass slide have these little probes. Each probe will measure the expression of one gene. And you can see the RNA for the gene in the two different samples will be hybridized to this. And then after hybridization and wash, you shine color onto the slide. You'll be able to see based on the color for whether some gene is higher in the normal condition or the cancer condition. For example, if it's dark, it means that this gene is not expressed in either conditions or expressed pretty lowly. Whereas if it's yellow, this gene is highly expressed at similar level, but in both conditions. But if it's green, this is a gene that is highly expressed in the normal cell, whereas if it's blue, it will be a gene that's highly expressed in the cancer cells. And so this is really fantastic. People can really use it to measure expressions of hundreds or thousands of genes at a time. And so many groups started adopting these type of microarrays to do expression studies of the condition they are interested in. Around the same time, there were also commercial solutions. So initial cDNA array was generated in a lab. The PhD students or postdoc would create those spotted cDNA arrays, but between conditions, sometimes you have very significant artifacts. And so there were industry solutions. And so in this array, there could be 6 million probes measuring all 20,000 genes. And because it's industrial manufacturing, the results were much more reproducible. And so people were, these are kind of more expensive, but people started using this for measuring expression. And I remember this is a 1999 study, seminal study done by Todd Golub at the Dana-Farber Cancer Institute, where I'm a faculty. So basically, there were two types of leukemia. One is a acute lymphoblastic leukemia. This is called ALL. The other is AML, acute myeloid leukemia. These two types of leukemia pathologically look quite similar. So sometimes it's hard to distinguish them for diagnosis. However, the treatment and outcome could be different. Therefore, reaching a correct diagnosis is quite important for treatment and for the patient. And so in this study, Todd Golub and his colleagues collected a number of ALL patients samples and AML patient samples. So each column is one patient sample. And then they profiled RNA on this commercial microarray to look at the expression of thousands of genes in each sample. And by looking at the differentially expressed genes, they were able to identify gene markers, for example, that are much higher expressed in ALL compared to AML. And they can also identify genes that are highly expressed in AML. So the goal is that in the future, if you have a new patient from pathology, maybe the doctor is having a difficult time telling these two cancers apart, you could profile the patient tumor or the blood sample with the microarray, and based on the expression of these marker genes, really reach a much more accurate diagnosis and give the patient the right treatment. And so this really kick off precision medicine. People are doing a lot more expression profiling experiment. And so if you look at gene expression in 2020, there are assays such as RasoSeq or Luminex assay. Basically, these are technologies that can profile the expression of about 1,000 important marker genes at very, very low cost. When the first Affymetrix microarray were invented, it cost close to $2,000. Then it dropped to $1,000 and then to like a few $100s. But now you can see here with RasoSeq or Luminex, you can do this with a few dollars per sample. And so with this type of technology, for example, recently the Broad Institute has a connectivity map project where they use this Luminex approach to profile over around a million different profiles of many different cancer cell types being perturbed in different conditions by either gene perturbation or a drug, a treatment. And then they can measure the expression of 1,000 genes. After they get the data, they can use the publicly available gene expression profile where people measure the expression of all 20,000 genes, and then use the 1,000 genes they measured to computationally impute the remaining expression. And then they can get really kind of a genome-wide expression signature of how the cell would respond to a particular perturbation. So these are really very useful resources for people to look at drug or gene effect. In addition, people are now developing single cell gene expression techniques. So instead of looking at a bulk tissue, single cell expression technique can look at within a complex tissue whether there are different cell types. For example, in the brain, there are different components, and also in the heart and in liver, there are so many different cell types. And so this is a recent study that was published last year in Jay Shenduri's lab at University of Washington, where they took 15 different fetal organs and 121 different samples, and using a technique that they developed called siRNA-seq. It's a kind of a split pool approach, which we will discuss later in this course. And using this approach, they were able to sequence over 4 million single cells. It's really mind-boggling. It's over 4 million cells. Each cell, they can reach hundreds and thousands of genes. So it's like a tremendous data matrix. And based on that data, they can cluster the cell types and also cluster the different tissues and organs, and then figure out how different type of cells in different organs are expressed and how they interact with each other in development. And so I hope you can see that the technology for gene expression also have evolved a lot, and with it there are a lot more data generated, and also computational biology is really essential for making sense out of that data.